Hello, my name is Dhriti Kuram.
Hello, I'm Aniya.
And I'm Aaron Powers. In this presentation, my team and I will investigate the abilities of a polyextremophile in an extraterrestrial environment and determine the application of the results on human space exploration.
Extremophiles are organisms that can survive in extreme conditions. Our project was to research an extremophile and then determine the ideal location to send the extremophile for its survival in space. To begin with, we will observe how they adapt and react to new environments. Finally, we will discuss our discoveries and research and how it could be used by humans for future research and the development of new technologies.
Our background research began with examining the Deinococcus radiodurans, a polyextremophile that is able to withstand dehydration, extreme heat, freezing temperature, as well as high doses of radiation, making these bacteria the most likely to survive in environments outside of the Earth. The Deinococcus radioduran is able to use oxygen to produce energy from organic compounds and reproduces using binary fission. However, the most fascinating characteristic of this bacterium is its ability to repair chromosomal damage and resist environmental stress that could be lethal to other organisms. An example of this would be radiation. This extremophile is able to absorb about 1.5 million radiation absorbed doses before losing basic function. After conducting research, our team concluded that exposing the Deinococcus radiodurans to extremely high levels of radiation in space would obtain the most beneficial results for improvements in space exploration.
We also determined the ideal location to send Deinococcus radiodurans was Jupiter's icy moon, Europa. Europa's surface consists of a solid icy sheet, and beneath the surface, experts believe, is an ocean of liquid water and slushy ice. Europa also has a thin atmosphere that is primarily composed of oxygen. The presence of water on Europa has made many wonder if life could survive there. However, the high radiation levels on Europa makes life seem impossible for most organisms. Due to Deinococcus radiodurans' resistance to radiation and DNA reparation, we have concluded that they would be the ideal organisms to survive on Europa.
This leads to the goals of the project. How are Deinococcus radiodurans able to resist intense radiation, and how can the radiation resistance help future humans when exploring Europa? With knowledge about the bacterium and the selected environment, this project would assist space exploration by observing changes in a life form outside of Earth, as well as changes we can make in the future to help humans while in space.
When exposed to radiation, the organism undergoes oxidative stress, where there is an imbalance between the reactive oxygen species and the antioxidant capacity of the cell. For example, high doses of radiation damage the internal biomolecules, which induce double-strand DNA breaks, or DSBs. The bacteria, however, have high oxidative stress resistance and are also able to repair broken DNA. The organism follows a two-step DNA repair mechanism that allows Deinococcus radiodurans to completely reassemble its radiation-shattered chromosome from hundreds of short fragments. The first phase is a process named extended synthesis-dependent single-strand DNA annealing. Newly synthesized complementary single-strand DNA extensions appear to anneal so that DNA fragments are joined together, forming long linear intermediates. The second phase involves REC-A protein-mediated break repair. REC-A-dependent homologous recombination is conducted to become circular chromosomes with DNA patchworks of double-strand DNA blocks, connected by those synthesized before and after radiation exposure. In total, the bacteria are able to combat environmental stress factors by constantly repairing DNA breaks.
If we were to send Deinococcus radiodurans to Europa, the ideal location would be the Pool crater. Images from the Galileo spacecraft showcase bright white rays stemming from the crater. These rays may indicate the existence of freshwater in fine ice particles, in contrast to the darker brownish areas around the crater. The possible presence of freshwater is attractive for sending Deinococcus radiodurans because, although they can function without water, it is ideal for survival. The extremophile's ability to survive in colder temperatures gives it an advantage in the environment. The bacterium is most radiation resistant in temperatures around -79°C, allowing the extremophile to survive in Europa. Although these cold temperatures are not ideal for growth, it pushes the extremophile to react and adapt to Europa's environment.
A gene editing process called CRISPR can be used to create DSBs, which could then be used to change the genetic code of human cells. After replicating the Deinococcus radiodurans DNA and allowing it to conduct reparation processes with the use of the REC-A protein, the DNA can then be inserted into the double-strand breaks of the human DNA, allowing for an increase in radiation tolerance to help astronauts survive on places like Europa. Along with this, after observing the bacteria, we can see if the Deinococcus radiodurans had evolved in any way due to the environmental stress. This could help predict the changes in humans as they explore the moon, as well as see what other life forms we could send into space.
A few of our highlights from the NASA SEAS program this year was being able to tune in for the Astrobiology speaker series, our exposure to the amazing research and opportunities, as well as interacting with our helpful and collaborative peers. The challenges we had to face were working virtually, limited resources on our topic, as well as the theoretical concepts.
Our team had an amazing experience with this internship. We enjoyed working with each other, and this opportunity solidified our interest in space science.
We appreciate the time you have spent listening to our presentation. Thank you.